Welcome to Breast Cancer Breakthroughs. One of the most influential things that we've learned about breast cancer is that your cancer can change and become resistant to treatments over time. Treatment resistance is a huge area of focus for researchers who are working day and night to try to find more options for patients whose tumors have become resistant to treatment. But did you know that treatment resistance and other changes in a tumor are often caused by genetic mutations? When we think about genetic mutations in breast cancer, we usually think about the kind of mutations that you might be born with, like mutations to the BRCA1 and 2 gene that could increase a person's risk for developing breast cancer and other cancers. But some genetic mutations can occur in a tumor after your breast cancer diagnosis or even during the course of treatment. But luckily, researchers are staying one step ahead with targeted treatments. In today's episode of Breast Cancer Breakthroughs, we'll be talking about a big advance in staying one step ahead of breast cancer by monitoring for certain genetic mutations and treatment options for patients whose tumors have changed. We'll speak with Komen Scholar Dr. McDonnell, who discovered one of these tailored treatments in his lab, and former grantee Dr. Aditya Bardia, who helped to bring these treatment options to life for patients. If you're wondering what is a genetic mutation, check out our video linked below. Last year, the FDA approved elacestrant, or Oserdu, to treat metastatic estrogen-receptor positive breast cancer with mutations in the estrogen receptor gene, which is called ESR1. In this episode, we'll be referring to this as ESR1 mutations. This approval was a long time coming for Komen Scholar and breast cancer researcher Dr. McDonnell, whose lab discovered elacestrant.
It's a great story, and it's a story that really highlights the ingenuity of some of the trainees in my lab. But the story didn't start a few years ago. It actually started about 25 years ago when I was interested in why is it that cancer cells become resistant to, at that time, the best endocrine therapy was tamoxifen. And we thought at the time it was just because tamoxifen was a good drug, but not a great drug. Let's go make better estrogen receptor inhibitors. And one after the other, the field was making these estrogen receptor inhibitors, testing them in tamoxifen-resistant disease, and they were all failing. And in fact, my last count when I wrote an article on this recently, there were 9 drugs that were evaluated for efficacy in patients who had failed tamoxifen. And so rather than just wanting to do the same thing over and over again, we decided to actually see, was there another way that we could actually inhibit the estrogen receptor in tumors? And the idea was to develop small molecules that would actually cause degradation of the estrogen receptor and then test them. I won't go through all how we got to them and things like that. It was a lovely science project published in a fantastic paper by the two of them in about the year 2000. And they found a drug. And the drug bound to the receptor, twisted the receptor into a shape that led the cell to believe that it was a denatured protein. And the protein was then thrown into the cell's trash can. And the estrogen receptor was gone.
After the initial discovery of drugs that could get rid of the estrogen receptor in breast cancer cells, which are also called SERDs, Dr. McDonnell's lab ran into some roadblocks. They were working with a few companies to move this drug into clinical trials, which is how we test new drugs for effectiveness and safety. These companies lost interest in developing this type of drug, and eventually the group needed to start from scratch with brand new molecules. They ended up with a molecule that was originally unsuccessful for treating hot flashes, which intrigued Dr. McDonnell's research team.
So what they found was, in a clinical study, was that as you increase the doses of this hot flash, of this treatment, you actually increased the number of hot flashes, not decreased them. And so Suzanne Wardell and Eric Nelson, who are 2 trainees in my lab, basically love weird pharmacology. Okay. And so they got their hands on some of this drug, and within 2 weeks, they had figured out the mechanism. And it turns out the very mechanism that was probably responsible for the failure in hot flashes was the very reason that it worked in breast cancer. Because what it did was, it did the same thing as our original drug. It bound to the receptor, it twisted the receptor into a conformation and that was denatured, and the cell threw it into the trash can and was gone.
Finally, by 2017, Dr. McDonnell handed the new drug to a pharmaceutical company who started a phase 3 clinical trial of the drug for breast cancer in 2019. And this trial was called the Emerald trial. We spoke with Dr. Aditya Bardia, a lead investigator on the Emerald trial, and he told us a little bit about the gap that the Emerald trial was seeking to fill for patients.
Yeah, absolutely. For a patient with metastatic hormone receptor positive breast cancer, the standard was endocrine therapy plus a CDK4/6 inhibitor first line. And in the second line setting, the drug that was used was fulvestrant, which had modest activity in the post CDK4/6 setting. And also patients were getting the medication as an intramuscular shot. So it was very inconvenient given that it's given as a shot twice a month for the first month and then once a month. So the unmet need was a better endocrine option in this setting. And second, something that's oral. So the Emerald trial was designed to address this unmet need. Can we bring an oral and more effective endocrine-based options in the post-CDK4/6 setting? So patients with metastatic ER-positive breast cancer who had received prior CDK4/6 inhibitor were randomized to elacestrant, an oral selective estrogen receptor degrader, versus endocrine therapy of physician's choice, which was predominantly fulvestrant in this setting. The trial had two primary endpoints: progression-free survival, all comers, and progression-free survival in patients with detectable mutations in the estrogen receptor. And we can come back to this point. The reason this was designed this way was we know in the second line setting, the mechanism of endocrine resistance is heterogeneous. There are a subset of tumors that are still ER pathway dependent. There are a subset that are not. So can we identify the tumors that are ER pathway dependent? And the use of ESR1 mutations was a surrogate marker, because if a tumor has estrogen receptor mutations, it's likely still being driven by the estrogen receptor. And so that was an important endpoint in this clinical trial. In general, elacestrant is very well tolerated. The rate of treatment discontinuation was very low with elacestrant in the clinical trial. The most common side effect that's seen is nausea, but it's usually low grade, not enough that you need an anti-nausea medication, but more like if some patients take ginger or some other strategies, or with food, and usually it's not a problem. It can also cause upper GI discomfort. So the use of an antacid or proton pump inhibitor, or with food again, could be considered. Outside of that, it does not have major side effects, not much hot flashes or muscle aches, joint aches. And given that it's oral, does not have the inconvenience of the intramuscular shot.
The results of the Emerald trial were positive. In patients with an ESR1 mutation, 26.8% of patients still had stable cancer at 12 months, compared to only 8.2% of those who received the standard of care. This is incredibly exciting because it's the first new hormone therapy to be approved in more than 20 years. And it offered a pill option to combat the significant problem of resistance to hormone therapies. After patients with metastatic estrogen receptor positive breast cancer receive their first line of therapy, some patients' tumors still rely on estrogen to grow, but some do not. If you're interested in learning more about how estrogen and the estrogen receptor drive tumor growth, you can check out our video linked below. We asked Dr. Bardia to explain the significance of an ESR1 mutation.
The key point regarding ESR1 mutation is that it's acquired. So when a tumor is inhibited by an aromatase inhibitors lower estrogen. The tumors can become resistant to aromatase inhibitor by developing a mutation in the estrogen receptor. And when there's a mutation in the estrogen receptor, the tumor is estrogen independent. That switch, which was dependent on estrogen, is constantly on. So you can bring the estrogen levels to very low or even zero. The tumor is estrogen independent and continues to grow, but it's still ER dependent. So drugs like aromatase inhibitors don't work in that setting because they work by lowering estrogen. But drugs that directly bind to ER, like elacestrant, would work. And that's why this was an important setting where this drug was developed.
As Dr. Bardia mentioned, the ESR1 gene mutation is something that you acquired during treatment with aromatase inhibitors, which means that it's not something that you're born with, like a mutation to the BRCA1 or 2 gene. We asked Dr. McDonnell to explain the difference.
For the most part, When we talk about a patient with a BRCA mutation, we're talking about somebody who has a genetic mutation. That is, that they were born with this mutation and they are predisposed because of that mutation to develop cancer. That's basically what BRCA is. Now, there are some tumors that can evolve to also acquire BRCA mutations, but they're not the ones we're focused on when we talk about BRCA mutant patients. But the ESR1 mutations are different. You're not born with those mutations. These mutations are somatic and they occur only within the tumor. And they occur only in the tumor where that tumor has been put under phenomenal selective pressure. So let me give you an example. The breast tumor, the estrogen receptor positive breast tumor really wants to use the estrogen receptor signaling pathway. It's learned how to do it and it wants to do it. And then we come along with the aromatase inhibitor and we take away the gas, take away the thing that it wants. Well, it says, well, I'm so addicted to the estrogen receptor signaling pathway, I'm going to figure out another way to turn that on. And so what happens is there are mutations that occur in every cell every day that die away because there's no selective pressure for them. But now, when you take away you add the aromatase inhibitor and you take away the estrogen, suddenly there's a selective pressure for these, and they grow out. So that's what you're talking about, evolution, an evolution of the tumor. Whereas in the BRCA, it's not. You're just born with that.
You might be wondering, how would I know if my breast cancer has an ESR1 mutation? Well, the good news is ESR1 gene mutations are extremely rare in early breast cancer. Dr. Bardia explained to us how ESR1 gene mutations are detected, which uses a blood test called a liquid biopsy.
In terms of how to test, because it's an acquired mutation, if you test the primary tumor, you would not detect ESR1 mutation. It's something that's usually detected by a blood test. So when a patient has disease progression on AI CDK4/6 inhibitor, at that time, if you do a blood test and do plasma-based genotyping, that's how ESR1 mutations are detected.
We've heard that not only can your tumor change during treatment, but your treatment, like the CDK4/6 inhibitors Dr. Bardia mentioned, can also influence how well you respond to subsequent drugs. Further analyses from the EMERALD trial found that the length of first-line CDK4/6 inhibitor treatment was associated with better response to elacestrant. While progression-free survival improved with duration of CDK4/6 inhibitor treatment with elacestrant, there was no difference with those who received the standard of care. Dr. Bardia also shared that a single test may not be enough to detect an ESR1 mutation during treatment. We asked him about when patients should ask their doctors about being tested for an ESR1 gene mutation.
After the first-line setting. So after disease progression and first-line therapy, it should be considered. And then something that, cost issue aside, if feasible, should be done serially because it can increase over time. So even if the first test doesn't show, does not mean that the next test would not show it.
The story of how elacestrant was brought from bench to bedside is an incredible illustration of how research discoveries are brought to life in the clinic to make a difference for patients. But ESR1 gene mutations are not the only mutations that can be targeted with drugs. Dr. McDonnell explained to us that there are other exciting advances in the field of drug development that are targeting other mutations in breast cancer.
The idea of actually targeting mutant proteins that is not that new, if you want. So it turns out that there are mutations in many proteins that give us constitutive activity. And one is PI3 kinase. Okay, if you have mutations in that enzyme, it's constitutively active, it drives the growth of the tumor, and PI3 kinase inhibitors have shown efficacy and are efficacious in patients with late-stage metastatic breast cancer. We still haven't quite got the right drug combinations yet, but they work. There are mutations in the EGF receptor, not in breast cancer, but in lung cancer. And again, we've developed drugs to target those. So I think that, and RAS is a classical one as well. The mutations in RAS that lead to activation, and they're targeted with specific molecules that only hit that mutation. So the nice thing about those is if they only hit the mutation and they hit the mutant protein, it is likely that the toxicities associated with those drugs are going to be considerably less. Because think about it, the mutant protein is a different protein. It's a different protein than the native protein.
Dr. McDonnell mentioned one breast cancer gene mutation in particular, which is PI3 kinase, that's had some really exciting breakthroughs in the last couple of months. Capivasertib, or TrueCap, was FDA approved in November for patients with advanced estrogen-receptor positive, HER2-negative breast cancer with mutations to the PI3CA or PI3 kinase, AKT, or PTEN genes. We also heard some really exciting results from the INAVO120 clinical trial at the 2023 San Antonio Breast Cancer Symposium, also known as SABCS, that found that a new drug called inavolisib can lead to a 15-month progression-free survival benefit compared to 7.3 months in the control group for people with estrogen receptor positive breast cancer with a PI3 kinase mutation. You can learn more about the results of the INAVO120 trial in our last episode of Breast Cancer Breakthroughs, which covered highlights from the San Antonio Breast Cancer Symposium of December 2023. Thank you all so much for watching. Here at Komen, we are committed to bringing you evidence-based information to empower you as you navigate your journey with breast cancer. Don't forget to sign up for reminders for Breast Cancer Breakthroughs. We've linked the homepage below. And finally, if you enjoyed today's episode, please take 2 minutes to fill out a short survey, which is linked in our description below.